Okay, so we have seen this in our previous assessment. Okay, just a quick revision on microscopy. Okay, this is the microvilli, basically the enfoldings of the cell surface membrane that creates this finger-like projections that you can see here, right? So only found on the surface, belongs to the gut lumen. Okay, so we have the intestinal epithelial cells obtained using a scanning and a transmission electron microscope. So that means that they require us to go back and make sure we know what are the features of the scanning electron microscope and transmission electron microscope. All right, so for the first one, okay, with reference to figure 1.1, okay, state how it is possible to distinguish between a scanning electron microscope and a transmission electron microscope. All right, so the point that I mentioned here is the one that we can see. When they say reference to figure 1.1, that means you must refer on what you can see from the images itself, okay? So from here, right, what we can see, okay, in A, okay, A is basically the image from the scanning electron microscope. Okay, I'm just going to write that down. Right, this is the scanning electron microscope. On the right is the transmission electron microscope, the micrograph from transmission electron microscope. All right, and apart from what I've put in here, A is from the scanning electron microscope because the image is showing external factors, and B is showing the internal structures of the cell. Okay, that's one of them. Other points that you can also consider is that for the scanning electron microscope, you can only see the surface of the cell, yeah, the outer layers of the cell, surface of the cell, right? Also shows 3D, okay, images of the cell. All right, and then they only show the external, which is what I've already included there. And then for this one, right, and then for the transmission electron microscope, okay, you can see that it's showing, not only shows the internal layers, the image are okay, in 2D, the images are in 2D, all right. And we can see the cross section, meaning you can see the inside of the epithelial cell. Okay, you can actually see the contents of the cell, which allows us to see contents. Oops, I just write that. To see contents of the cell. Right? Or the internal parts of the cell, same meaning. Now, I know that in the lecture slides, okay, that I've included that, you know, some of the image actually have colors. So as you can see in the exam, we can't really use that. Okay, besides the images, the color images that you see are all false coloring images, yeah? Okay, which is actually generated by the computer that we use to view the micrographs of the scanning and the transmission electron microscope. All right, so moving on. So approximate length of microvillus is 1 micrometers. So outline the method. So that means we have to explain how we are going to estimate the magnifications. Oh look, there's a hint there. Magnifications of the images shown in figure 1.1. Now because they give you hints there, they're asking for magnifications and we're going to be estimating. So that's why I want to emphasize on, okay, the idea that we need to take a few measurements, okay, a few measurements of the microvilli. Okay. So first, I mentioned the formula. So everybody should know the formula of magnification by now. M is equals to I over A, right? Now, what we need to do is we need to take the multiple, okay, measurements of the length of the microvilli from both figures, okay, from both figures on the left, from figure A and from figure B. So we're going to get multiple, okay, measurements of the length that we can see here, all right? And then calculate the mean. Okay, again, this is magnification. So we're going to be calculating the mean to get the image length. So what we're measuring here, that would be your image length. Okay, so this is image length. Now, because we actually take multiple measurements and we have the mean of the image length, and then what we can do is that we can divide this value to the actual length, which is mentioned here. Approximate length of a microvillus is 1 micrometers. right? And that's how we can get the magnification once we substitute it in the formula. Okay, the idea of taking multiple means is what I think they want us to show them. Next, okay, the role of an intestinal epithelial cell is the absorption of glucose from the gut lumen from the lumen into the circulatory system, which involves different membrane transport proteins. Okay, the events occurring in intestinal epithelial cell during the absorption of glucose are summarized in figure 1.2. So this is figure 1.2, and they actually give us the summary, right? So we've got the image here, right? And then let's look at the summary together. Point number 1. Sodium ions are removed from the cell by active transport. They actually tell us it's being removed by active transport through a transport protein known as the sodium-potassium pump. So I've indicated that with this blue star. So that's what's currently happening here. Okay, there's active transport. Okay, off. Sodium out, right? But at the same time, potassium ions are taken in. Okay, so this actually, okay, where we have antiport. Okay, so you can take note, antiport. When one substances or molecule is transported out, another one is transported in. Next, okay, because of the results of what's happening here, it decreases the concentration of sodium ions in the cell compared to the gut lumen. So now the concentration of the sodium ions here in the cell is lower compared to the gut lumen here. Alright, so there's reduction. Okay, fine. So we got that noted. Glucose molecules are transported together. Okay, it means transported together, right, with sodium ions into the cell from the gut lumen. So I've indicated that with this star. So this is what's currently happening here. So we can see that sodium ions and glucose are transported together into the lumen. Okay, and then last but not least, glucose molecules are transported out of the cell into the tissue fluid down a concentration gradient. So I think here it's kind of obvious that's obviously diffusion, or to be correct, facilitated diffusion, because it requires transport protein. So in this case, what I have indicated here, right, you can see that the glucose are transported out down its concentration gradient into the tissue fluid, right, and it enters the capillary. Okay, so that's the scenario that is summarized by the diagram. So they want us to answer the next question. Active transport involves water-soluble substances such as sodium ions and potassium ions, and the use of ATP to provide energy for their transport through carrier protein. So that's basically them helping us to define what active transport is already. So again, if you were asked to define, okay, this would be a good note for you, right? Okay, ATP. Okay. Now outline other features. They want us to mention other features of active transport that is not mentioned in this definition here. So the answer is, we know, point number 1, substances are moved against the concentration gradient. Take note, right? Second, they are moved through carrier protein. And when that happens, what happens in the carrier protein is that the carrier protein undergoes conformational change in its shape as it transports those materials through its structure. Okay, the idea of this conformational change. Okay, for 2 points, 2 marks. Okay. And next question. Glucose molecules enter the cell through a membrane protein. Suggest why glucose molecule needs to be co-transported with sodium ions when it enters the cells through the membrane protein. Alright, so we went through the summary here. Okay, note that for the first 2 summary, it tells us here that sodium ions are removed first by active transport, lowering the concentrations of sodium ions in this structure, right, mentioned here. Okay, so when there is lower concentration of sodium ions, it's allowing for this process to next occur. So essentially, the purpose of this epithelial cell is it's wanting to absorb glucose. How is that useful? Alright, so that what brings us the idea that when there is lower concentration of sodium ions in the epithelial cell, okay, sodium ions will then be able to diffuse into the epithelial cell. Now, when it diffuses into the epithelial cell, that would be via facilitated diffusion, because we're talking about it's passing through a membrane protein, right? So, through a transporter protein, membrane protein. And when it does that, it carries along glucose, right, because it's co-transported. Now, glucose is going against its concentration gradient, all right? Now, we want to make sure that glucose are able to be transported in, okay? There's already a lot of glucose, but we're transporting them together against its concentration gradient, okay? Because there's already a lot of them here, okay? And then the idea that this ensures that, so this ensures that the glucose will then be able to be fully removed from the gut, and so that it can enter the epithelial cell. Okay, so we want to ensure that all of the glucose are going to be removed from the gut, enters the epithelial cell, so that eventually it can get, okay, transported out into the tissue fluid and then into the capillaries, as mentioned here, down a concentration gradient. All right, so meaning there's already high concentration of glucose here. So transporting glucose here with sodium, okay, through co-transporter protein, allowing the glucose to be moved against its concentration gradient. Right? Okay, moving on. Explain how microvilli increase the uptake of glucose into an intestinal epithelial cell. So the idea, the foldings actually increases the surface area. For what? So that a lot of this transport protein can be embedded within the structure. Then, why is that good? So more transport of glucose can occur. So what does that mean? So if you have, okay, you can see that we have these transporter proteins, right? Okay, on the epithelial cell, on the microvilli. So essentially, by having more foldings, so what you can do is that you can allow more of those transporter protein to be embedded within. So imagine like I'm drawing them hypothetically here. So having more inward foldings, more epithelial cell, having these co-transported proteins, so more glucose will then be transported in. Okay? Alright, next, last but not least, stem cells are located in the walls. They divide by mitosis. Make sure that you know what mitosis is. Basically, okay, when there is nuclear division causing the cell to divide, producing genetically identical daughter cells. So I want us to suggest and explain the importance of mitosis by stem cells in the small intestine. So make sure you know what stem cells are. So stem cells are basically undifferentiated cells which has the ability to continuously divide by mitosis. Alright? So, alright, we know, okay, let's revise back on mitosis, okay, that mitosis has several roles. They have roles in growth of cells, allowing cells to grow bigger, right? It is involved in the repair of tissues when there's tissue damages. It is also involved in cell replacement when there is, injured, damaged cells. They replace the cell, right? It is also involved in asexual reproduction, all right? And whereas the undifferentiated cell is essentially for stem cells that I mentioned earlier. Okay, so the answer that would be relevant here is the idea that, when we see, okay, this small intestine always working, right? I mean, there's bound to be, time where the cells would need to be replaced or the tissues might be damaged, for example. So the relevant one that we can actually include in our answers here would be 3: growth, repair of tissues, and cell replacement. And how I'm going to write that in my answer is the idea that it allows damaged cell to be replaced, see, replacement, okay, when needed. And then after that, Okay, we need to link the role of stem cells. Stem cells have the ability to divide and then differentiate into intestinal cells. So they can become new intestinal cells, which produces identical epithelial cells of the intestinal cells, which then replace damaged cells. So cell replacement, allowing it to repair the intestinal tissues. And then mitosis will then aid in the growth of these cells in the small intestine. So that's how I actually use all the points of mitosis into my answer. Okay.